Hello friends, welcome to the channel. In the last lecture we discussed about the introduction of microbiology, and today in the second lecture we are going to study about the bacteria and its cell structure. So bacteria, they can be simply defined as ubiquitous microscopic single-celled prokaryotic organisms that have varying shape. This means that ubiquitous means they are found almost everywhere, whether it is the air, it is soil, or anything else. They are microscopic, means they cannot be seen by unaided eyes, they cannot be seen directly, and very oftenly, we make the use of microscopes to see them. Single cell, these are unicellular structures, means they are made up of only single cell. And prokaryotic. Prokaryotic means they are devoid of a true nucleus. That is why they are prokaryotic organisms. Now the bacteria, they can be considered both harmful also and helpful also. They are considered harmful because they cause a number of diseases in human beings and animals, and they can be considered helpful because they help in the formation of many foods such as the probiotic foods like curd, cheese, etc. The bacteria provide the first line of defense in our body by staying inside our respiratory tract or the digestion tract or on our skin, and therefore they help against the invasion by any foreign invaders. These are very simple organisms, but they have a very developed cellular structure. As you can see, the bacterial cell, the outermost layer of the bacterial cell is called mucilage. This is the mucilage, and sometimes we also call it as the glycocalyx or the capsule. The mucilage layer is made up of mucopolysaccharides and the glycoproteins. It helps the bacteria in extreme situations. Inside the mucilage lies the cell wall. Now cell wall is a very important structure because the cell wall provides the structural integrity to the bacterial cell. It is responsible for the rigidity of the bacterial cell. Now the cell wall is a relatively porous structure. It is made up of peptidoglycan, that is the polymers, means these peptidoglycan, these are the polymers of N-acetylglucosamine, N-L-glucosamine, and L-acetic muramic acid. Glycan is responsible for the rigidity of the cell wall and for the determination of the cell shape. However, the primary function of the cell wall in the prokaryotes is to protect the cell from the internal turgor pressure that occurs due to the much higher concentration of the proteins inside the bacterial cell. Now inside the bacterial cell lies the cell membrane or the plasma membrane. The cell membrane is a semi-permeable structure, means it allows the entry of only selective molecules. All the molecules cannot enter the cell directly or they cannot leave the cell. Only selective movement of or the selective entry of the molecules inside the cells is allowed. Now according to the fluid mosaic model, the cell membrane is a bilayered structure that consists of two layers of the phospholipid molecules with the globular proteins associated with them. This cell membrane acts as a barrier between the cell and the environment. That means it helps the cell sense its environment or interact to its environment. Also, the cell membrane is a elastic structure, means if you are going to pull it with a micro instrument like some needle, first of all it will get pulled up and after that it will retain its original shape, just like in rubber band. Bacterial cytoplasm, there are numerous organelles present which perform different functions like the ribosomes and polyribosomes, which help in the protein synthesis. You can see the respiratory enzymes also. Now bacteria power up its metabolism process using the membrane bound respiratory enzymes, which capture the chemical energy and transduce it by pumping the protons and sodium ions across the cell membrane. There is the presence of nucleoid. The nucleoid contains the bacterial genetic material. This is a very irregular structure and it lacks a nuclear membrane. There is the presence of glycogen also, which acts as a reservoir for carbon and as a energy source. Now there is some free RNA also present in the bacterial cytoplasm. There are several filamentous structures like the pilus and the flagella. The pilus is found in the gram-negative cells. We will discuss further what are gram-positive and gram-negative cells. This pilus helps in the attachment of the bacteria to its surface. Now there are more lengthy structures like the flagella. The flagella helps in the locomotion of the bacteria. The outermost filament of this flagella is the outermost portion of this flagella is known as filament, means this portion. Now the flagella is attached to the bacterial cell with the help of a hook, and the downward portion of the flagella is known as the basal body. On the basis of the gram staining techniques, the bacterial cell walls have been classified into gram positive and gram negative. If the cell walls retain the crystal violet color, the bacteria are known as gram positive bacteria, and if the cell walls they retain the pink color, the bacteria are known as gram negative bacteria. The gram positive cell walls have teichoic acids in them. Now this is a very important question which has been asked several times in many of the examinations, that teichoic acids are found in the gram positive bacterial cell or the gram negative bacterial cell. So you should say it is found in the gram positive bacterial cell wall. The negatively charged, and therefore it gives the overall charge to the gram positive bacterial cell as negative. Now the gram negative cell, in the case of gram negative bacteria, there is the presence of an external layer of the lipopolysaccharide molecules. These lipopolysaccharide molecules are also negatively charged, and therefore the gram negative cell walls they also have a negative charge. Now the gram positive cell walls, these are comparatively thicker as compared to the gram negative cell walls. Now the common examples of bacteria are like E. coli, which is most commonly found in the water that we drink or any other aquatic deposit. We know the disease cholera. This is caused by Vibrio cholerae. We know about botulism, which is a kind of food poisoning. This is caused by Clostridium botulinum. Now there are diseases and infections. Now there are various infections and diseases that are caused by bacteria, and we will study them in our next lectures. That is all for the day today. Thank you.